Good morning, everyone. Welcome to Breakfast Club, for which we are also welcoming two really special guests, one of whom you've seen before, but are, I know, excited to see back. We have our herpetology curator, Dr. Raina Bell, and our herpetology collections manager, Lauren Scheinberg. Hi, both.
Hello. So in the collections, we preserve things usually in three different ways. Most of the specimens you'll see today are wet specimens, and so they're in alcohol, either fixed with alcohol or formalin preserved, kind of like embalming the body, but ultimately stored in 70% like drinking alcohol. Another way that we preserve specimens is skeletal material. And so this is an articulated snake in like a archival deli container. But some of our specimens, especially ones that aren't in great condition or maybe don't have the best associated data, will skeletonize like this. And then a third way I wanted to show, which is one of my favorites, is cleared and stained. So we don't do this as much anymore, but this was a way of preserving a specimen where you take off the skin and then you use a variety of chemicals and heat to clear the flesh. And so all of the muscles are still there, they're just clear. And then we use two different dyes. One dyes the bones red and one dyes the cartilage blue. So it's a really nice way to see all of the internal structures and all of the bones without it kind of falling apart, which sometimes it'll do if you just preserve it as a skeleton.
Okay, great. That was really beautiful, and thank you for showing us.
So behind the offices and public floor at the Academy, on every floor we have these collection rooms. And so the one that we're showing here is our main wet collection room. But we do function as a library for scientific research. And so I send out, in a normal year, I would send out about 1,200 specimens and 1,200 associated tissues to researchers around the world. And so when I do that, I take whatever specimens I have that they want out of the jars, I wrap them in alcohol-soaked cheesecloth, and then I triple bag them and send them in the mail. So they're sent like that all around the world for researchers to use.
And I'd love to see what you pulled today, if you're ready for that.
Yeah, let's do it. Let's move on.
Okay.
Try and get as much data as we can from each of these specimens. And so that includes the data that we take when they're collected, but we do get all of the data from them. We preserve them. We take tissues for DNA work. And then occasionally we will look at their stomach contents. And so snakes swallow their prey whole and then slowly digest their food. And so sometimes, this is going to be a rough one to take out, but this is a rattlesnake. You can see its little rattle here, but then we also have its stomach contents, which I'll just show in the jar because it's going to be too hard to get out. You can see this is what came from its stomach, and it is an almost complete collared lizard.
Wow.
And it is roughly the same size, at least not the length, but in width of the rattlesnake itself. So we'll often take out, or researchers will take out the stomach contents to see what they were eating when they died. And then in another example, we have another rattlesnake, a local rattlesnake, with more digested stomach contents. And so in here is just kind of some digested fluff from, I think, a rodent.
Using museum specimens has been a really important way for herpetologists to learn about the diets of snakes and how they change throughout the different life stages within a species, how different species that are living in similar habitats eat different prey items, so kind of similar to Marcel's research with the caecilians. And then also things like the relative size of a meal relative to the size of the snake predator. So all kinds of really interesting research that has come out of using museum specimens. And like Lauren said, really just trying to get like the most information that we can out of each individual specimen that we have. And this is just a really cool video that's sort of the opposite extreme of snake diet research, which is some researchers from Southern California who have been studying interactions between rattlesnakes and the different species of kangaroo rats that they predate. And they have this really cool setup where they have infrared high-speed videos. And so they go and find snakes that are out at night in an attack posture, waiting for prey items to come by. And then they set up their cameras and see what happens. So there's a whole series of these if people want to watch more. And this is just a short one to give you the idea of how amazing these interactions are.
Whoa.
Yeah. So, you know, we think of like the rattlesnakes as being these really amazing predators, and of course they are. And this is just to show that even with all their specializations for being really amazing predators of small rodents, that it's actually a really challenging thing to pull off. We realize that when people think about snakes, they often think about rattlesnakes, and then at the other extreme, these really large-bodied boas and pythons. And you might not realize that we also have boas that are local to California and to the Western United States. And they are very small and gentle in comparison to what you might initially think. So we have two species, the rubber boa and the rosy boa. And I don't know which one Lauren wants to show you first.
I have a rubber boa. I don't have
Okay.
a rosy boa specimen out, but I have a rubber boa because I think that they're great. They're a local species. I can find them like in the Bay Area. They're docile. They're not flashy at all. And they're wrinkly and brown and kind of cute. This is a rubber boa face and body. They mainly eat pinky mice, like rodents. And so their tail is really stumpy, and they'll sometimes, and it kind of resembles a head, so they'll use that as a little decoy. And so often they'll have really scarred tails from being attacked with their tails. But in general, they're smooth, brown, wrinkly, local friends.
Yeah, and one of the things that's really neat about boas, and we couldn't find a good photo of this, we were looking, but they kind of like a whale retains parts of the pelvis from when whales were hippo-like mammals living on land. Boas retain vestigial hind limbs as well. And they're just like a really tiny femur and a really tiny pelvis that aren't connected to the spine anymore. And they're positioned on either end of or on either side of the cloaca, which is the opening through which everything happens at the back end of a snake. And so there's one on either side, and they tend to be a little bit bigger in males, and they use them in courtship.
Thank you so much for being here today. And again, for all the work it took to put together the tour itself, the virtual tour, which people loved. So again, Lauren and Raina, thank you so much. That was so cool. And viewers, thanks for being here, and we will see you next week. Bye.
Stay safe, everybody.